Our next topic is organogenesis. As you can guess from the name, organogenesis, this is the process of organ formation that occurs once the embryonic germ layers have been laid down. At this point, the embryo really begins to increase in size, and since the nervous system is the first thing that starts to develop in most vertebrate organisms, we're going to focus on the development of the nervous system. So just be aware, we're going to talk specifically about a process that has its own name related to nervous system development, but there are other processes for other systems that we're not going to talk about. So we're not going to do kind of an all-encompassing look at organogenesis. We're just going to look at this one specific thing that kind of exemplifies how organogenesis works. So this process is called neurulation. This is the process of forming the nervous system. Now, in order to get to this, we need to change the orientation of our embryo a little bit. So let me just orient you here within this embryo. This is a much later embryo than we've been looking at. So here we have our three germ layers. We've got ectoderm, mesoderm, and endoderm. And we have the structure called the notochord right here. So this is the dorsal side of the embryo. This is the ventral side of the embryo. The notochord right here is that connective structure or support structure that is derived from mesoderm. So right here. Now this notochord is going to do a thing called induction, which just means it's going to signal nearby cells to do something. And in this case, the notochord is going to induce the nearby cells of the ectoderm to thicken and form a thick region called the neural plate on the dorsal side of the embryo. And that's this kind of blue-green region here. This is the neural plate. So induction is just a process where one cell or group of cells is going to stimulate nearby cells or influence nearby cells to differentiate in a specific way. So in this case, it's the mesoderm cells of the notochord are inducing the ectoderm cells of the dorsal side of the organism to form the neural plate, which will eventually give rise to the central nervous system. So if we just look at what's going on that dorsal surface now, we're not showing anything here except the dorsal surface, and in teal we have the neural plate. So the neural plate is this region in the embryo that rolls up. So if you can picture this, it's kind of folding downwards, and the sides are rolling up like two waves that are crashing into each other right here. This is why the dorsal hollow nerve cord that we find in chordates is hollow. It's because of the way this process of neurulation works. So we have this structure that folds inwards and the sides fold over, and eventually those sides are going to pinch closed. So you have a flat dorsal surface right here, and then below that, just ventral to that, but still on the dorsal surface of the organism, you have a hollow tube that is ultimately going to become the dorsal hollow nerve cord. The anterior end of this structure is going to fold over more and develop into the brain, and we'll talk about that when we get to the nervous system. And the rest of that tube is going to remain a hollow tube, and that's going to become the spinal cord. So that is the process of neurulation, where the neural plate forms due to induction from the notochord. It folds over and pinches off. So we end up with a dorsal hollow nerve cord, and the anterior end of that is going to specialize into the brain. This process explains something that I've mentioned a bunch of times but seems really weird. It's that the nervous system is derived from the ectoderm, which, if you think about that at first, it doesn't make any sense. You have the kind of innermost tissue of the body, the thing that is most protected inside your body, is covered by the most other layers of cells. Well, that stuff is derived from the outermost embryonic germ layer. How does that happen? It happens because of this process of neurulation, where that ectoderm layer folds inwards, and the tissue that ends up on the inside as the result of that folding is going to become the nervous system. An interesting note is that this is a common site of developmental problems, or at least it was common prior to us figuring out the importance of folic acid during embryonic development. So there are a number of spinal cord and central nervous system disorders, the most common of which is called spina bifida. The idea with these is that this folding and closing and pinching off doesn't occur properly in the case of a folic acid deficiency. Folic acid is also known as vitamin B9, and if you have ever been pregnant or know anyone that's been pregnant, you know that vitamin B9 is an important prenatal supplement. Basically, there's a lot of like prenatal vitamins out there, but really if you ask like a doctor, what they say is that unless you have some kind of weird thing going on with your diet where you're not eating something, you're not going to have any vitamin deficiencies, but we're going to put you on B9 anyway, just to be sure, to avoid these neural tube defects, these problems where the neural tube doesn't entirely close and separate during neurulation. So that's the importance of folic acid, vitamin B9 during embryonic development. Now, overlapping with neurulation and also subsequent to neurulation, we have lots of other processes that are broadly under the heading of organogenesis, and these involve a lot of cell migration. So cells are not staying in the same place during embryonic development. They're moving all over the place within the embryo. Some of these are fairly local and involve local signaling and regulation, and some of them are fairly long-term moves and fairly long-term regulatory networks across the embryo. So the cells of the neural crest, for example, which is part of that neurulation process, if I could just go back a minute here, these cells right here, so this is the neural crest, this part that's a little bit like grayed out right there, it just refers to like the crest of the wave, and some of those cells end up on the inside, but not part of the neural tube. So those cells of the neural crest actually migrate throughout the body. They become nerves, so they become part of the peripheral nervous system. They become the teeth. They become some of the bones of the skull. So there are these weird little pockets of tissue in your body, such as your teeth, that are derived from ectoderm, even though it seems weird and counterintuitive that that's the case. It's because they're derived from these neural crest cells that are migrating throughout the body. Another group of cells that are really important for organogenesis are called somites. Somites are blocks of mesoderm that are lateral to the notochord, so they're on either side of the notochord from front to back in the body. So here you could see very clearly you have these blocks of tissue right here. Those are somites in this particular embryo. Somites are often involved in making structures that are repeated through the body or segmented structures. So if you think about things like ribs and vertebrae, and the associated muscles like the intercostal muscles that are between the ribs, all of those different structures are derived from somites, these blocks of mesoderm that are involved in generating repeating or segmented structures. So at this point, we're going to jump ahead in the process. We're not going to do anything more specific with organogenesis, but we'll finish up today by looking at human development.